Hello, everyone. Today we're going to be going over the liver. So, we're going to be talking about morphogenesis, engineering processes, pathogenesis of the liver, current strategies, and how to develop a better liver. So, as you can see on the left, this is an overview of liver development in mice, and then the right shows a table comparing how long each stage takes in different animals. As an overview of liver development, it begins with endodermal patterning, followed by hepatic competence. Then hepatoblasts form and differentiate into hepatocytes and cholangiocytes, followed by a period of maturation. Endoderm patterning is when the fore, hind, and midgut are separated. And then within the ventral foregut is where the hepatic competence is gained. Hepatic induction begins with a dual BMP FGF signal, where hepatoblasts then migrate and expand into the STM, forming the liver bud on the ventral wall of the foregut. As the hepatoblasts expand, TGF beta is secreted by the portal vein, which drives cholangiocyte differentiation while suppressing hepatocyte differentiation. Hepatocyte differentiation occurs further away from the portal vein, where HNF and OC2 counteract the TGF beta. The last phase in liver formation is liver maturation. Hepatocytes gain function based on their specific location within the lobule. Hepatocytes closer to the central vein have different functions such as glucogenesis in the periportal zone, where there's more oxygen. At the same time, cholangiocytes form bile ducts, which are then surrounded by hepatocytes. At this point, the lobules and the bile duct of the liver are formed. This slide shows the molecular pathways involved in all the stages on the left and the cellular pathways on the right.
Engineering processes are critical to the early phases of liver development. In order for the hepatoblast to invade the surrounding STM during liver bud formation, they need to transition from epithelial to mesenchymal cells. This delamination is characterized by a decrease in ECA-adherent expression and consequently cell adhesion. At the same time, the surrounding basal lamina needs to be degraded by matrix metalloproteinases produced by the STM and hepatoblasts. Without either of these processes, the hepatoblasts do not invade the STM and the liver does not properly develop. For the actual migration, the surrounding laminin-rich ECM acts as a permissive factor. If the hepatoblasts are deficient for the ECM binding protein beta-1 integrin, then they are unable to migrate into the STM and the liver is underdeveloped. Following liver bud formation, the growth and patterning of the liver is highly dependent on mechanical factors. Between E10 and E15 in the mouse embryo, the liver becomes highly vascularized, which consequently leads to rapid growth. The mechanotransduction pathway has been implicated in this extensive growth. Beta-1 integrin molecules are co-localized on the surface of vascular endothelial cells with VEGFR3. Blood flow through the vessels distorts the surrounding ECM, which is sensed by the beta-1 integrin molecules, leading to the activation of VEGFR3. This signaling leads to the downstream production of HGF, which leads to extensive hepatoblast proliferation and differentiation into hepatocytes. Cell ECM binding then helps pattern these hepatocytes into their characteristic hepatic cord structures, which can be seen in the figure on the right. Binding through the alpha-3 beta-1 integrin heterodimer naturally induces this cord-like formation. When the heterodimer is inhibited, the hepatocytes are unable to form the characteristic cord structures. Cholangiocyte patterning is also dictated by mechanical factors. Shear stress and substrate stiffness have been shown to manipulate hepatoblast differentiation patterns. More specifically, tensional forces and low substrate stiffnesses promote hepatocyte differentiation, while compressive forces and high substrate stiffnesses correlate to cholangiocyte differentiation. These results translate to the in vivo patterning seen since the hepatoblast interacting with the portal vein would be experiencing high substrate stiffnesses due to the portal mesenchyme secreting ECM and compressive forces from surrounding cell mass, leading to cholangiocyte differentiation seen around the portal vein.
Cirrhosis is a progressive disease developing slowly over many years. The buildup of scar tissue can eventually stop liver function, ending in the need for a liver transplant. Building the hepatic epithelial system is a complicated morphogenic process, and given the amount of molecular and cellular regulation needed, it is no surprise that the smallest impairment in this process can result in acute and chronic liver disease. Fibrosis is the encapsulation and replacement of injured tissue by a collagenous scar and results from the perpetuation of the normal wound healing and regeneration response, which then results in continuous connective tissue production and deposition, which is known as fibrogenesis. The pathogenesis of fibrosis is still not fully understood. With persistent damage, hepatocytes lose their regeneration capacity and large fibrous scars replace them, leading to a chronic condition. Following multiple injuries and/or exposure to inflammatory cytokines, hepatic stellate cells undergo the transition from an inactive to activated state. Activation is a pivotal event in initiation and progression of hepatic fibrosis, characterized by cell proliferation, contraction after transforming into myofibroblasts, and a major contributor to collagen deposition, ultimately leading to liver fibrosis. Cirrhosis is a liver disease known for its distortion of the hepatic vasculature, advanced stage of liver fibrosis, and being a leading cause of morbidity and mortality. Hepatocytes play a complicated role in cirrhosis and are targets for most hepatotoxic agents. Chronic liver disease either promote apoptosis or trigger compensatory regeneration of hepatocytes. Damaged hepatocytes release reactive oxygen species and fibrogenic mediators, induce activation of HSCs, and stimulate the fibrogenic actions of myofibroblasts. The profibrogenesis effect of TGF-beta is complicated. Involving multiple aspects, the primary effect of TGF-beta is to stimulate activation of HSCs. To the left, you can see a normal liver. The blood runs through the hepatic sinusoids where endothelium, which rests on loose connective tissue, allows for extensive metabolic exchange with the hepatocytes. Blood is collected by terminal hepatic venules, which discharge into one of the 3 hepatic veins. On the right is a cirrhotic liver. Advanced myofibroblasts that derive from hepatic stellate cells and portal or central vein fibroblasts proliferate and produce excess ECM. Blood is directly shunted from terminal portal veins and compromises the liver function.
In severe cases, liver transplant has become the only definitive cure, but the availability of donor organs has not been able to keep up with the rising demand. A substantial number of patients die on the waiting list of liver transplants. Therefore, regeneration or repair of liver tissue has become increasingly important to study. The biggest problems that researchers face when trying to rebuild the liver include sourcing of liver cells. They are quite limited in number, variable in quality, and unable to be expanded in vivo. If used incorrectly, they can also cause severe fibrogenic responses, which does more harm than good. Another challenge stems from the lack of knowledge regarding the liver's ECM and architecture. For this very reason, artificial scaffolds are not as robust as using decellularized matrices. Since the location of hepatocytes dictate their function, trying to recreate scaffolds as close to the natural one is crucial in order to build a fully functioning liver. Early on, researchers tried to do hepatocyte transplantation where they introduced more hepatocytes into diseased livers and found that this did not really help improve liver function and actually cause more fibrogenic responses. Although implantable engineered tissue is quite novel and holds promise, it's only applicable in small hepatic mass loss situations as it cannot produce large amounts. Du et al. Studied the interactions between the hepatic epithelial and non-epithelial cells and found that they were crucial to the development of the liver architecture. So using combinations of hepatic specification factors, they could use lineage reprogramming to make hepatocyte-like cells. Yin et al. Showed that the stellate cells played a major role in causing the decline of liver regeneration and also causing liver regeneration. Although there are more of these developmental techniques, they have not been applied in most natural and artificial scaffolds. Through the implementation of these developmental techniques in the future, we can hope to see more robust regeneration of the liver.
With a better understanding of liver morphogenesis and fibrosis pathology, now we're going to address some of the challenges when it comes to developing a therapeutic strategy. So first, we need to stop the progression of liver fibrosis and reverse hepatic fibrosis, and also find a way to modulate the primary factor of activating hepatic stellate cells, TGF-beta signaling, in order to reduce the amount of fibroblasts and ECM deposited in liver fibrosis. So what do we know so far? Well, we know that the prolonged activation of hepatic stellate cells and collagen deposition leads to liver fibrosis. In liver fibrosis, as seen previously, many of these signals are released, and thus there's a lot of molecular network signaling pathways that induce liver fibrosis. From these signals produced, TGF-beta is the strongest activator of fibrogenesis, activating these hepatic stellate cells into myofibroblasts, and thus also increasing collagen production. Without the degradation of collagen, nor the deactivation of these hepatic stellate cells, we cannot reduce the progression of liver fibrosis. But we also know from previous literature that the regression can be done by either having these hepatic stellate cells go into senescence or cell death, apoptosis. Now, in order to combat this TGF-beta signal, we've also identified an important regulator of TGF-beta called caveolin-1, which will help us modulate the TGF-beta signal produced in order to activate these stellate cells into myofibrillets and create collagen. So what is caveolin-1? Caveolin-1 is a structural protein of the plasma membrane. It creates this invagination, thus capturing TGF-beta1 and releasing these anti-apoptotic signals, thus an inducer of hepatocyte proliferation. It plays a lot of liver functions. It plays the regulation of a lot of liver functions, but most importantly, it's this release of anti-apoptotic signals that we're going to use in order to modulate TGF-beta1. So with this knowledge, we have developed a strategy where we're going to introduce hepatocytes to overexpress this important modulator caveolin-1 in order to reduce this release of TGF-beta signaling. And thus, by reducing the expression of TGF-beta, we hope to reduce the number of hepatic stellate cells that are activated, and thus reduce the amount of myofibrillets created, and also reduce the amount of ECM deposited, and be able to break that down in order to restore that regeneration function. So how are we going to go about this? Well, first, we're going to develop some characterizations tests. First, we need to develop these hepatocyte-like cells through using induced pluripotent cells. We're going to reprogram them with the known hepatic specification factors and use immunohistochemistry staining in order to identify whether or not we have actually developed hepatocyte-like cells. After that, we also want to test whether we can alter the genome of these hepatocytes in order to overexpress caveolin-1. So using our known strategies, we know that we can attach caveolin-1 gene, CAV1, to an ubiquitous promoter albumin and be able to overexpress caveolin-1. We're gonna check ourselves using immunohistochemistry by tagging it with GFP and using Western blot in order to identify the expression level of caveolin-1 once we edit the cell. So now that we're able to characterize our cells, now we're going to move into our experiment. Here, we want to first identify whether we have an association between caveolin-1 and TGF-beta expression. So what we're going to do is we're going to culture the hepatostellate cells in a TGF-beta environment and incorporate caveolin-1 into the media. By doing so, we're going to see the effects of caveolin-1 on the TGF-beta expression and also the activation and proliferation of hepatic stellate cells. Through immunohistochemistry, we're going to track the proliferation and activation and also the ECM deposition. We're going to track collagen, laminin, and alpha smooth muscle actin in order to see how much ECM is being produced. We expect to see an increase in caveolin-1 expression reduces the activation of hepatic stellate cells and the proliferation of it, and we also should see less collagen production. Once we have established this association, we move into our in vivo model. We're first going to introduce these hepatocytes with this overexpressed caveolin-1 to reverse liver fibrosis in mice. So first, we're going to take cirrhosis-induced mice and transplant our edited hepatocytes. As with the timeline shown below, we will sacrifice the mice first at 1, 2, 3 days, and then go on a weekly basis at 7 days, and 15 and 30. By using this sacrificing timeline, we're hoping to capture the whole regeneration process, which occurs usually from 5 to 7 days, and then capture how it affects the lung after post-regeneration. So how are we going to track that? We're going to first, once we sacrifice the mice, we're going to take out the liver and take its mass. We're also going to stain it to identify proliferation and activation of the hepatic stellate cells. And we're also going to quantify it because we also want to know whether or not we're able to restore the liver function as well by tracking the enzymes that are critical for many metabolic functions in the liver. And thus, through this in vivo model testing, we hope to find a reduction in the myofibroblasts, a reduced collagen deposition, and increase of protein proteases that will degrade this ECM in order to restore our hepatocytes. Now, with this model, we have several limitations. Now, we have designed this timeline, sacrificing timeline, but this is based on the assumption we have this 5 to 7 days post-injury. Perhaps we have to add in more time points and perhaps sacrifice more frequently or less frequently. So depending on our first initial study, we'll be able to tell whether or not we're able to fine tune what time points we can observe the best changes. And also, caveolin-1 is bound to right now an ubiquitous promoter. We have seen above that TGF-beta signaling is very important for post-injury and liver regeneration because it activates these stellate cells. So we don't want to just kill all the TGF-beta in the environment. So we might have to employ a dosage control mechanism in order to modulate whether or not there's a certain level of expression that could lead to the right amount of modulation that will reduce the amount of activated cells and be able to switch from a fibrotic pathology back into our hepatocytes and liver regeneration process.